Hi everyone, my name is Gabriella Venable and I am a PhD student at Duke University with Brian Hare's research group. We are launching a large-scale PhyloPsy collaboration on the evolution of shy boldness and are hoping to include a diverse set of species. Anyone with access to a population of at least 7 individuals of the same species with an interest in our study is invited to join. You can contact us by hitting the Request to Join button, followed by the Contact Lead button, or by emailing either me or Brian. With this study, we hope to address the following questions. 1. Do temperament traits evolve in response to the same selective forces hypothesized to impact cognition? For example, does the complexity of a species' social system or diet predict reactions to novel objects, food, or social agents? 2. Is temperament related to brain size? Comparative work suggests brain size is related to cognitive performance, but is the same relationship true for temperament traits? Currently, it is unclear if temperament traits are related to measures of brain size. If unconstrained by brain size, does that mean temperament traits have the potential to evolve faster or more readily than cognition? 3. How is temperament related to cognition? It has been hypothesized that temperament could modulate cognition in a variety of ways. For example, changes in social tolerance and emotional reactivity are proposed to impact cognitive evolution. Additionally, links have been suggested between temperament and learning speed. Temperament has even been proposed to prevent or allow the expression of certain cognitive abilities. Thus, temperament evolution could perhaps be a driver of cognitive evolution. To examine these questions, we will use a shy boldness test battery to complete a large-scale comparative study of primate and non-primate shy boldness. We will use phylogenetic comparative analyses to examine 1. The relationship between a species' ecology, temperament, and cognition while controlling for phylogeny. 2. Whether there is a link between measures of brain size and shy boldness. And 3. The relationship between shy boldness and measures of executive function, i.e. working memory and self-control. For our large-scale comparison of temperament, we will be measuring shy boldness, i.e. reaction to novelty, which is a core temperament trait. Our team of collaborators will measure what subjects prefer to approach to characterize each species' avoidance/attraction to novelty. To do so, we will be using a very simple paradigm. Essentially, subjects will be allowed to choose between 2 stimuli. In the experimental condition, subjects will be given a chance to approach either a novel or a familiar stimulus. In the control condition, we will measure subjects' approach in response to 2 familiar stimuli. We are trying to categorize species' overall shy boldness, and therefore we need to test responses to a range of novel stimuli to rule out context specificity. To test the link between temperament and cognition, we will largely rely on data reported in 2 previous publications, a study of self-control from McLean et al. 2014 and a study of short-term memory from Many Primates et al. 2022. For species not included in these existing datasets, we invite collaborators to consider adding to these comparative datasets by using the same methods reported in these previous publications. A few materials will be necessary to complete these temperament tests. You'll need a set of food bowls or another food receptacle, preferred food rewards, and novel objects. Ideally, these novel objects will be about 1/3 to 1/2 the size of the subject, have at least 4 colors, red, yellow, green, and blue, be made up of at least 4 different materials, and have a shiny component. For our experiment, we used felt, paper plates, hot glue, palms, ribbon, and shiny duct tape. Our novel objects are single-use, but your species-appropriate stimuli may be reused across trials. Just make sure to maintain novelty across subjects. In other words, do not let subjects observe ongoing trials with other individuals, etc. You will also need 4 flavors of Jell-O that are different colors, 4 face masks, 4 long-sleeve shirts, 4 scents, and a way to video/audio record trials. If any of these stimuli are not appropriate for your species or are not novel for your study population, please contact us. We may be able to include some variation in our methods that works with your species. Subjects will be tested individually in a familiar location. In other words, they should be familiar with everything other than the novel stimuli. In each trial, the test stimuli are placed in one of two locations about 4 to 5 body lengths from the subject and equidistant from the subject's starting point. For example, the entrance to a testing enclosure. A trial begins when the subject is at the starting point and ends after the subject either approaches both choices or after 10 minutes elapse. Each block of trials will consist of two conditions. First, the control condition. The subjects are presented with two familiar stimuli, familiar food and/or familiar experimenters. Second, the experimental condition. The subjects are presented with a familiar stimulus, i.e. familiar food and/or familiar experimenters, and a novel stimulus. To control for side bias, the novel stimulus will be placed on the side the subject approached first during the control trial. In each trial block, the control will be run followed by an experimental trial. Our measures are latency to approach, or the time it takes to approach, and the order in which the subject approaches the two stimuli. An approach is scored when a subject touches or places its head or grasping arm over one of the stimuli while on the same surface as the stimuli. However, a different but comparable criteria may need to be developed for your subjects based on the specifics of your study population, particularly the physical arrangement of their testing space. If possible, the ideal design will be to test subjects of each species in either 6 sessions of 4 trials or 12 sessions of 2 trials for a total of 24 trials. Each experimental trial will be preceded by a control trial so that half of the trials are control and half are experimental trials. The order of conditions presented for a test battery is novel object, then novel food, and then novel social agent. We welcome our collaborators to collect more data using a counterbalanced trial order, but to keep our data comparable, ask that they first complete trials using this order of novel stimuli. 6 lemur species have been tested at the Duke Lemur Center using the method described here. Here are some example videos from the pilot study. First, let's look at a video of a control trial for a novel object. In our trials with the lemurs, the starting point for novel object and novel food is the entrance to the testing enclosure. The subject is allowed 10 minutes to approach both stimuli, i.e. the 2 bowls of familiar food. Should the subject fail to approach both stimuli, the trial block is aborted and can be repeated on a different day. The animal passes the entrance to the testing enclosure, which is our starting point. As you can see, the subject first approaches the distal bowl and then the proximal bowl of food, after which the trial ends. In the following experimental trials for this subject, the novel object would be placed where the distal bowl of food was in the control. Again, control trials for both novel object and novel food were run the same way in our pilot study. Now we will see an example of an experimental trial for novel object that was done with a different species. The animal enters the testing enclosure. In this case, the object is the further choice and the familiar food is the choice closest to the camera. The subject approaches the familiar food first and then carefully approaches the novel object. In each trial, the animal is given the chance to eat all of the familiar food. We also try not to interrupt the animal while it is actively engaging with the novel object. Now we will see an example of an experimental trial for novel food. The familiar food is in the distal bowl and the novel food is in the proximal bowl. Here we see the subject approach the familiar food and then the novel food. It is then scored whether the individual in fact eats the jello. As you can see, in this trial the animal approaches the novel food but does not eat it. Now we will see an example of the control trials for novel social agent. These have a different setup from the novel object and novel food conditions because in our testing context we are not able to enter the enclosure with our subjects. We use tables because our study population shows the highest motivation to interact with experimenters in this way, i.e. off the ground. To assure that the 2 choice locations were roughly equidistant from the subject's starting point on each trial, we used a bowl with a small treat on the back table to anchor the subjects before each trial. Once the subject places his head over or reaches into the bowl on the back table, the trial begins, and the experimenters each offer an identical food reward. During the session, to make salient the social agency of the experimenters follow the subject with their eyes and face direction. In this example, the subject first approaches the experimenter on the left. Therefore, in the following experimental trial for that trial block, the experimenter on the left will wear the novel social agent costume. Now we will see an example of the experimental condition with a novel social agent. Note that the novel social agent will put on their mask while the subject is eating from and next to the back bowl at the starting position. In this example, we see yet another species. The subject approaches the back bowl and the novel social agent puts on their mask. At the same time, the experimenters also offer their food. For our novel social agent trials, this back bowl of food is our starting position. We see the subject approach the novel social agent first, and then the familiar experimenter. Once the animal takes food from the experimenter, that experimenter removes their hand. In order to join this study, hit the Request to Join button followed by the Contact Lead button, or email the email address on the screen. If you are interested in the study, but unsure that the methods will work for your study species, please reach out. It may be possible to include variation in the methods for your species. Please also contact us if you have any other questions and/or concerns. Thank you for watching and listening to this video. More details can be found in our project documents folder or on the YouTube description. Please reach out if you have any questions. We would love to have you participate in our large-scale collaboration.